Today, my group and I present diseases in seed and development for our biology SL. We are from Group 9, Kuliah 5. To expect from us. Today, in our presentation, we will be giving a brief introduction of members, the presentation of the main topic, the introduction of the interviewees, the interview clip itself, and the presentation of a subtopic, and ending it with our conclusion. So, who are we? We are Amalia Khadija Sofrina, Haris Hazmi, myself Kritisha, and Nurul Aisha Farhana. What is seed and development? It is a pivotal process in the life cycle of an angiosperm that is initiated by a process of double fertilization, which leads to the development of the embryo and the endosperm.
Okay, Assalamualaikum. I am Amalia Khadija. So I am going to introduce our interview, which is Prof. Dr. Uma Rani Senior. Okay, so who is she? She is an expertise in seed science and technology PhD in Seed Science University of Reading, UK, and have a bachelor in agriculture science, UPM.
Assalamualaikum and good morning to my teammate and especially to our special guest, Professor Dr. Uma Rani, anak perempuan senior. So hi Prof, how are you today?
Hello everyone, I'm fine, thank you.
We will talk about deeply in diseases in seed development and the way we can prevent it from happening. My very first question is, what are the common diseases of monocot and dicot seeds?
When you ask the question, common diseases in monocots or dicots and so on, basically there are so many different types of diseases, and these are being caused by either fungal, bacteria or viruses, and they are quite specific actually.
So I'm going to ask you the next question, which is what steps should one take to prevent diseases from occurring in seeds?
You have to keep checking for the symptoms to see if something is happening, and then we treat the plant.
So I'm going to ask you the next question, which is how do we identify plant diseases in seed stage?
The seed, when you take the seed, basically the seed will grow into seedling. We can identify diseases at the seedling stage, but there are also many ways where we can test the seed itself. We can do a sampling. There are methods, for example, called blotter method, agar method, visual observation method. A visual observation, we just observe under microscope.
So madam, as for the plant disease, is there any eco-friendly way to control it?
Today, people use the word management. The method can be as simple as you go to your plant, and if you see there is some insects, for example, you see snails. Instead of killing the snails, you just pick the snails out. If you have a huge farm, this may not be the easiest method. Biological can means you have plants that deter certain types of insects that bring diseases. You can have things where you can just keep your farm clean.
The viruses and diseases from the animal that are contracted by human. Does this happen with plants as well?
We do not know because some mutations may happen somewhere, right? But usually plant diseases are not anything that humans or animals will contract.
Okay, so if I'm not wrong, that is all for our question and answering session with Dr. Uma Rani Senior.
Assalamualaikum and hi. My name is Haris Hazmi and I will present to you the background of the disease in seedlings. There are many ways that seeds can be affected, such as the fungi, virus and bacteria. The first one is fungi. Fungi is the most common seed diseases. Why? We can see it very often on plants. The general effect from the fungi is they kill the plant cell. Fungi can spread through wind and water splash and also by planting materials. Most of these diseases are manageable. Virus is an intracellular pathogenic particles that infect seedlings. What that means is it is one of the pathogens that attack seedlings and plant. They have RNA single-stranded and needs a host to duplicate themselves, and they wouldn't attack unless the plant have wounds to enter. And the third one is simply bacteria. They are same as the virus because if the plant have no open doors or wounds, they would not simply penetrate the seedlings. They need several conditions to survive in the seedlings, such as optimum temperature, moist surrounding, and pH control.
So now I am going to talk about the symptoms of diseases in seeds. There are three things that can cause diseases, which is the first one is fungi, the second one is bacteria, and the third one is virus. Fungi. So the first one, you're going to find bird's eye spot on the berries. So if the plant has berries, has fruits, then you can see at the berries there will be a bird eye spot. Next, there will be leaf spots on the leaf. There will be spots brown in color. And then the third one is chlorosis, which is the yellowing of leaf. So the damping of seedlings, it is because of the fungi that affect the root system, causing the seedling to not be able to grow correctly. Okay, so next, bacteria. So the first one, it's almost the same with fungi, but this time it leaf spot with yellow halo. So like if you see at the leaf, there will be spots, but around it will be yellow in colors. And then the second one, on the fruit, there will be spots. And then crown gall, which is on the tree trunks or the twigs, there will be an abnormal growth of gall. And the last one is canker. Lastly, for virus, the symptoms are, the first one is mosaic leaf pattern. You can see at the leaf of the tree, there will be just like pattern, like mosaic like pattern. And then next is crinkle leaf. So if you see that your plant have crinkle leaves which it curl and does not grow correctly, and also sometimes it's blackened and turn brown in color. Okay, next one is yellow leaves. So your leaves that should be green turn yellow. And then the last one is plant stunting. You notice that your plant did not grow correctly. It's like stunt, which they do not grow as they should, and also smaller in size, even though you already planted for a long time. So that concludes the symptoms of diseases that can happen in plants.
Hi, my name is Aisha Farhana. So today I'm going to present the disease management. Okay, as for this topic, the farmers always use IPM method, which is known as Integrated Pest Management in handling the diseases. There are basically 4 methods in IPM, which is biological method, chemical method, cultural method, and also physical method. Okay, so as for the biological method, it is a method that controls pests such as mites and insects by using other organisms. For example, to control the aphid colonies from spread out, the farmers will put the ladybugs into their gardens. Next, move on to the chemical methods. It is a method that uses pesticides in killing the pests. For instance, chemicals such as herbicides are used to control or retard the weed's growth. Next, as for the cultural method, it is a method that reduces pest establishment, reproduction, dispersal, and survival. Okay, for example, the farmers will change the irrigation practice to reduce the pest problems, as too much of water can cause increase of weeds and also root diseases. Last but not least, as for the physical method, it is a method that kills the pest directly, blocking pests out or making the environment unsuitable for it. For example, the pests were knocking off of plants with a spray of water. So next, I will pass the presentation to Kirtisha.
Thank you, Aisha. Next, I would like to talk on the method of destroying this diseased plants or seedling. So the first step is to remove it from the ground itself, so that the other plants around it doesn't get infected. The second step is you either burn it or bury it. But burning has always been a traditional way. However, now, in this modern era, it is banned since it causes air pollution. So the next ecological way, which is eco-friendly, is to bury the diseased plant itself in a big pit that is far from other plants so that it doesn't infect anyone else.
Okay, so as for the disease transmission, it is not quite same for each disease cause. Like for fungi, it basically can be spread out by water, animals, insects, and also people. For example, fungal spores, which is very small and light, can travel through the air to infect other plants. Next, as for the virus, it physically enters the plant and are not spread by water or wind. For instance, insects that feed on the infected plants will transmit the virus to healthy plants when they are fed again. So lastly, as for the bacteria, it can be spread in several ways, such as insects, splashing water, other diseases plants, or tolls. They also can enter the plants through the tiny openings, either like through damage, cuts, or through the natural opens in the plants itself.
So I will continue the slides. So, identifying diseases in seedlings. The first one, we have International Seed Testing Association, or ISTA. They produces internationally agreed rules for seed sampling and testing, accredits laboratories, provides international seed analysis certificates, promotes research, disseminates knowledge of seed science and technology, provides training. So we can see that ISTA is an organization that have the authority to handling the seeds. All problems related to seeds will be referred to them. So the first method that we can recognize a disease is visual observation. This observation can provide quick information on insect, diseases, and mechanical damage to the seeds. This experimental process are the inspection of dry seed, so we can see the pathogen that affect the seedlings by change to the shape, size, or colorization of the seeds. The second one is blotter method. The blotter method is widely used for detecting fungi, which are able to produce mycelial growth and fruiting structure under the incubation condition available in test. And the third one is agar method, detects and identifies seed-borne fungi through colony characteristics. So this is the infographic about the methods that I just said before. This is the blotter method. Blotter method uses blotter paper that can absorb excess water. So seeds are incubated for a certain period of time on or between the blotter. And this is the observation experiment. We observe the seedlings and the plant itself to recognize the diseases. And the third one is agar method. Agar method used to identify the colony of fungi that grows on an agar like this. The detection of pathogens are more reliable than the blotter method because we can identify much accurate and quicker. So next, we need to prevent the disease from happen. The first one is to reduce the incidence of seedling diseases. Plant only top quality seeds by using the methods that we just said before. So the second one, seeds that germinate quickly and produce vigorous plants are more likely to survive the seedling stage. And the third one is plant the seed at the proper date. Planting too early greatly increases the risk of seedlings diseases.
To treat these diseases, it would be safe. Like you will be given antibiotics for a bacterial infection and antifungal for fungal diseases. In conclusion, the development of seed into seedling is a crucial process. Just like humans, they too need tender loving care. There are so many unique things about a seed, which are some still unknown and undiscovered in this world. Thank you for listening to us.